Welcome to Meiosis and some other stuff. You will learn about 7 things in this video: interphase, Meiosis I, Meiosis II, spermatogenesis, oogenesis, disorders caused by meiosis errors, and the reasons why parents differ from their children. So let's get to it. Meiosis I is preceded by interphase. During interphase, the cell performs its regular duties, making ribosomes, sending them off to the rough ER, making mRNA, making proteins, packaging stuff at the Golgi apparatus, etc., until the cell gets bored and decides to do meiosis. So the cell copies its DNA and proceeds with meiosis I. The first stage of meiosis I is prophase I. During prophase I, DNA strands coil around proteins called histones to form chromatids. The chromatids are then pinched together to form chromosomes. Each chromosome pairs up with its fellow homologous chromosome, and each chromosome trades parts of DNA strands with its homologous chromosome. This is referred to as crossing over. At the same time, the nucleolus disappears, and spindle fibers form, complete with handy centrosomes to carry them around. Next is Prometaphase I. During Prometaphase I, the nuclear membrane disappears, and the nucleoplasm within dissipates. Then one kinetochore forms per chromosome. Time for metaphase I. The spindle fibers grab onto the kinetochores and push the chromosomes into the center of the cell. Next is Anaphase I. The spindle fibers pull the 2 rows of chromosomes to opposite sides of the cell. And finally, telophase and cytokinesis. During telophase, 2 nuclear membranes form. Also, the chromosomes are split into chromatids and cleavage furrow forms. During cytokinesis, the cytoplasm divides. We now have 2 diploid cells. Now that we have 2 diploid cells, it is time to convert this into 4 haploid cells. And that is where meiosis II comes in. Meiosis II is the same as meiosis I, except that there is no duplication of DNA prior to it, and it applies to both of these cells. Watch what happens. Prophase II, Prometaphase II, Metaphase II, Anaphase II, Telophase II, cytokinesis. Notice that there are now 4 haploid cells. Each cell has half as much genetic material as a diploid cell. Now that you understand meiosis, which is just meiosis I and meiosis II in succession, it will be easy for you to learn how sperm is made. The process by which sperm is made in males is called spermatogenesis. During spermatogenesis, which occurs in the testes, a cell called the germ cell divides through mitosis to form another diploid cell called a spermatogonium. The spermatogonium then divides through mitosis to form a primary spermatocyte. This is still a diploid cell. The primary spermatocyte then undergoes meiosis to form 4 haploid cells. These cells then differentiate to form sperm cells. Oogenesis is the process by which eggs are produced in females. During oogenesis, a germ cell divides through mitosis to form an oogonium. The oogonium then divides through mitosis to form a primary oocyte. The primary oocyte then undergoes meiosis, but it is slightly different. During meiosis I, one of the resulting diploid cells gets most of the cytoplasm, while the other gets little and is called a polar body. Likewise, one of the haploid cells that results from meiosis II gets most of the cytoplasm. This cell is the egg, and because it is haploid, when it fuses with a haploid sperm cell, a diploid zygote is formed. Now I will discuss disorders caused by errors in meiosis. Two will be discussed in this video: Klinefelter syndrome and Down syndrome. A person with Klinefelter syndrome has an extra X chromosome. This is caused by nondisjunction, the failure of two homologous chromosomes to move to separate cells during meiosis I, or the failure of two chromatids to pass to separate cells during meiosis II. If this happens during spermatogenesis or oogenesis, then when the egg is fertilized, there will be an extra chromosome. In the case of Klinefelter syndrome, an extra X chromosome. A person with Down syndrome has an extra chromosome 21. This is caused by nondisjunction. To finalize this video, I'll explain two reasons why there's genetic variability between parents and their children. The first is this: during spermatogenesis and oogenesis, crossing over takes place during prophase I. Remember that crossing over is the sharing of genetic material between two chromosomes. This creates completely unique chromosomes. The second reason for genetic variability is the fact that when a sperm cell fuses with an egg cell, the sperm cell's chromatids pair up with the chromatids from the egg cell to create chromosomes. This means that the resulting zygote has chromosomes each with one chromatid from the father and one chromatid from the mother. This also means that the baby born will have chromosomes like this as well. And that's it. Two reasons why there's genetic variability. And all of this. But there is a mistake in this video. Remember that during interphase the DNA was copied. Now each cell initially starts out with 2 copies of each chromosome. So by prophase there should have been 4 copies of each chromosome. But if you look closely, that is not the case.